BY4 mock, it's the June 2015 paper, question 4. So here's a practical that most of you will have actually done, separating the pigments from photosynthesis in a plant. The blue-green pigment is chlorophyll A. State precisely where chlorophyll A is found in a photosynthetic cell. So hopefully you've got your photosynthesis notes and you're going, where do I actually find this stuff? So it's all to do with the light dependent reaction, and then can you find on these diagrams somewhere where chlorophyll A is going to be. So that's just in your notes. Here's a calculation, and it actually gives you the equation there. So calculate RF value of the yellow band and use the table to identify it. So what you need to do is look at these. Where's the yellow band there, and where's the solvent front there? And so with your ruler, measure that, measure the solvent front, put those into here, and how many decimal places should I use? Well, how many decimal places am I given there? And I'm usually going to follow the same style as I've been given. So I'm going to use 2 decimal places there. And that number that you come up with should be extremely similar to one of these in the table. You should be able to identify it. Part C. Some of the pigments were extracted and they've done this chart with them. So they're showing them different colours of light, which equals the wavelength, and they're measuring the absorbance. Unfortunately, the examiner is selling you a little bit of a dummy here with the name of this chart. You should know the name of the chart. You should have your notes there and go, yeah, I've got the charts, I recognise this, and I've got the names there. I'm not just going to copy this axis title there. I'm going to remember the proper name of the chart. Explain the results in the 500 to 600 range, so from there to there. And so we have got very low absorbance of the light. So the light is not being absorbed by these pigments. So where is the light going if it's not absorbed? And hopefully you realise why in this kind of green area. That's why plants, leaves look green, because reflection of the light. Explain the advantage to plants of having more than one pigment. Hopefully you notice from here that these are various pigments. It doesn't tell you which pigments are which. I'm just going to look at this wavelength there. Even though this pigment isn't absorbing much light, these other 2 pigments are. Here, this pigment is still absorbing light, and so on. And so we want to know the advantage to plants. So the first point might be describing the absorbance, the advantage at these different wavelengths of having 3 of these lines rather than 1. And then answer the question, the advantage to plants. What are plants going to do with this light that they've absorbed? So what are they going to do with it? A little bit of theory there. Which process do you know about that's going on that involves this light? So which process is going on that involves that light? What are plants going to do with it? Part D. Use your knowledge of photosynthesis. to explain the role of these pigments. So it's basically saying, okay, these pigments and photosynthesis, what's going on? Well, that is a rephrasing of the question, tell us about the light-dependent reaction. So tell us about the light-dependent reaction. And so all this stuff about light harvesting and light-dependent really is producing ATP. So it's all this kind of stuff, light-dependent reaction, and focus on the question, how do they actually produce ATP out of it? And it's a lot of space actually for 3 marks, so you should get those.